Well, hello everyone. Welcome to another episode of GU Cast. We are on tour. We are at the inaugural ProsTIC Preceptorship 2022. And this feels all wrong because normally Declan would be doing this, but today I'm joined by my guest co-host, no stranger to GU Cast and a great friend of ours, Professor Arun Azad, Medical Oncologist extraordinaire from the Peter MacCallum Cancer Centre. Hi, Arun.
Renu, thank you. Where's Declan?
Yeah. It's a long story. You can see him in the corner, right?
Why? I'm honoured to be here, but why?
I've been upgraded.
I always knew you had his eye on his job. I knew it. Yeah,
I have been working towards this.
We all knew you've trained. He's master and apprentice, and you're taking over now.
Well, he is on sabbatical this year, really. So I figure I should get the practice in while I can.
Yeah. What do they say in TV land? Never take a holiday because your guest host will come in, do a better job, and suddenly you've got you're out the door. So
Declan has a technique. He just takes his equipment and leaves.
Yeah, and I think what we need to do is see how many views this gets, and if it outrates Declan's previous ones, then I think the audience has voted, right? And it's just time to...
It might be you, though, Arun.
Time to ship. It won't be me, it'll be you.
But great to have you here.
Yeah, it's great to be here. It's an exciting day. It's a big day for prostate cancer, actually. Yeah. I mean, not just the ProsTIC Preceptorship, but there was also, just slightly off topic, there was today olaparib, PARP inhibitor, was actually announced PBS reimbursed for BRCA1/2 altered prostate cancer. That was a conveniently... there is a federal election coming up, as you know, so just a coincidence, of course, that this drug that we've known about with positive phase 3 data for 18 months is now on PBS, but that's great.
Exciting.
It's obviously great for prostate cancer patients. So we've actually had some, after a long period of time, we've actually had some approval or reimbursements for prostate cancer now in Australia through the PBS. So that's another one which is terrific.
Absolutely. I mean, we're waiting for the day when PSMA PET might be funded, right?
Yes.
That would be another... but, good news is coming fast.
Exactly.
So maybe this will happen in threes.
Yeah, well, who knows? Sooner.
Who knows? But I mean, it is a fantastic conference because, the material that we're hearing in the talks are amazing. The plenary sessions are really good. Tom Hope has been out there and, he's an amazing speaker on stage, isn't he?
He's like the American Hoff.
Yeah. But very engaging.
Yeah, he's a great speaker, and really, he's been terrific. I haven't actually had much to do with him before, but he's just been a terrific speaker. I mean, it's been really great talks. I mean, I think, ProsTIC and Peter Mac really is a world leader in prostate molecular imaging and Theranostics, and that really came home today. And Australia is more broadly. I mean, obviously, our friends Louise Smith and others, outside of Peter Mac as well, are also really at the forefront. And to see all the progress has just been terrific. It's
incredible. And Tom Hope's talk today about the upcoming clinical trials, really it was featuring Australian trials mainly.
Yeah, no, it's true. So I mean, and as well as, certainly all the investigator-led ones, or many of the investigator-led ones are out of Australia. Peter Mac and then St Vincent's in Sydney with Louise Emmett and her team. So it's fantastic for our patients and for Australian biomedical research.
And I think one of the most exciting things about all these upcoming trials is the translational side of it. It's often quite hard to find a clinician scientist, but this time or this weekend, I feel like I'm surrounded by them.
Well, you're one as well. You're doing a PhD in based on the Lutectomy trial samples, but that's actually a perfect segue for our. We've rustled up 3 guests today. We just found them in the lobby.
We found them in the lobby. Yep.
Just floating around, and we thought
they look like they needed to get something off their chest.
They look like and get them away from the bar and come up and do a podcast with us. You're absolutely right, though. The translational opportunities and actually opportunities to do more novel clinical trials to optimise outcomes from Lutetium PSMA are vital. So we've got 3 fantastic guests today who can actually elaborate on that. So
I think we should let them introduce themselves. Let them, you tell us a little bit about who you are and kind of how you came to be here and what your roles are.
Sure, Luc Furic. I've been working at Peter Mac for 6 years now. I've been named the head of Translational prostate cancer research at Peter Mac this year, so I've been in a role for 1 year. Very exciting role. Actually, at the preceptorship today, we've saw so many of my colleagues presenting what they're doing, and it's an amazing unified team of clinicians, nurses, radiochemists, and basic researcher like me, to bring these new discoveries, and do research and potentially bring new treatment to patients. The approval by the FDA of Lutetium PSMA as a treatment, just bring a new modality in the toolbox that we have now. And as researcher, I guess we'll have to see how this drug can be combined, how it affects tissue, and very excited to be here and to chat about all these opportunities.
Fantastic, Luke. And, I've got to say, we believe in multidisciplinary care at Peter Mac. And, I see you every week at our MDMs, getting ideas, giving ideas, and that's really what it's all about. So
And the free food, but anyway.
Yeah, the croissants. Is it worth getting up at 7 a.m. coming to work for?
They are.
Yeah, okay.
Sponsored croissants are the best.
Yeah.
I'm sorry, there's croissants.
We'll give you the details, Heidi. She's piped up. Why don't we go to Heidi next?
Yeah, we'll go.
I'm Heidi Fettke. I'm a postdoctoral research fellow at Peter Mac. I'm within the Translational Prostate Research Lab, and I spent the last 5 years working in liquid biopsies and identifying biomarkers for advanced prostate cancer treatment. My aim really is to improve outcomes by optimizing therapeutic approaches. I sort of stumbled into ProsTIC actually after I started here at Peter Mac last year. And it became very clear, I guess, that there's a real gap in the research at the moment for Lutetium PSMA therapy. There's no real genomics research going on, but there is a lot of liquid biopsy samples being collected via ProsTIC trials. So I'm really excited to take, I guess, my expertise into the Theranostics realm and try and use these liquid biopsies to identify biomarkers of response and resistance.
Amazing.
Yeah, that's no. We'll definitely elaborate on that. I've been very fortunate to be Heidi's PhD supervisor, and it was a really great experience for me to have her as one of my first students. And then we've got our third guest, Dr. Louise Kostos. So, Louise, do you want to give us a brief?
Hi everyone. I'm a long-time listener, so it's very nice to finally make an appearance.
Wonderful, Louise.
So I'm one of the medical oncologists at Peter Mac. It's now my third year working at Peter Mac, and I've just started doing a PhD in prostate cancer under the excellent supervision of Arun and Luc, and also Michael Hoffman.
Fantastic.
And my PhD is mainly focusing on trying to improve outcomes for men with metastatic castrate-resistant prostate cancer who are receiving radionuclide therapy. And part of it is, I guess, we'll probably talk about some of the mechanisms of resistance and reasons why Lutetium PSMA doesn't work in some patients, or why patients progress. And through a couple of early phase clinical trials, we're trying to evaluate novel combinations to try and overcome some of those mechanisms.
That's great. I mean, Louise, and just me also just say, I mean, I think it's fantastic to see, I think, medical research, has been often very male, especially in prostate cancer, very male driven. I think it's fantastic to see, three outstanding young female, researchers and clinicians at the table here, doing, really world-leading research. So that's fantastic to see. So just wanted to make that point. But
yeah, it's great to see more women out there doing this, isn't it?
Definitely. No, definitely.
For sure.
I mean, so if you set the scene, Renu, I think that, lutetium is a very active treatment. I think that was something that we talked about a lot today in the sessions, but it also came through that it's a treatment that not all men respond to, and unfortunately for a lot of men, it's not durable, the benefit, which is obviously disappointing. And so there's obviously many different ways that you could potentially try and improve that. And I think, two of the ways are, novel combinations involving lutetium PSMA, and then I think identifying biomarkers that help us better select patients and understand resistance to lutetium. So, I mean, I think that's actually a perfect scene for us to talk about some of the research that Luc, Louise, and Heidi do. So, should we start with the clinical trials or the biomarker work? Which one do you want to start with?
Biomarker work. Let's do that.
All right. Well, Heidi, perhaps we'll throw some questions your way first. I mean, I think that, one of the things that we know in prostate cancer, and you've been in the ctDNA, circulating tumour DNA space for the past 5 years now, and really an international leader in that space in prostate cancer. And we know that liquid biopsy is very important in prostate cancer because it's hard to get tissue, bones, hard to biopsy. And at the same time, we know there's a lot of heterogeneity in response to treatment, etc. So tell us about what's involved in getting a sample for ctDNA, what you do with it, and then what sort of data you get from that could help us understand treatment response and resistance.
So, just specifically cell-free DNA?
Yeah, cell-free DNA, yeah.
Okay,
yeah.
sure. So, we basically, our aim is to take serial liquid biopsies from our patients over the course of therapy rather than just a baseline. That really entails collecting peripheral whole blood samples from these men. We then shuttle it straight up to our lab. We process it straight away into plasma. So the plasma is the portion of the blood that contains this circulating cell-free DNA. So cell-free DNA is, everyone has cell-free DNA in their blood. However, people with cancer also have a portion of this DNA that is derived from the tumour shedding DNA into the bloodstream. And all this occurs at all metastatic sites, so that's one of the advantages of taking a liquid biopsy rather than a solid tissue biopsy. You're going to get a better, more comprehensive picture of a tumour. You're going to have DNA from a single blood draw that is derived from multiple metastatic sites. So once we do have our plasma that contains our cell-free DNA, we can extract it just using a kit. And then we move on to our optimised assay. So we've spent the last few years setting up a targeted panel sequencing assay where we look at mutations in 89 genes that are commonly mutated in prostate cancer. We're able to identify both somatic and germline mutations, which is really important, as you know, in the context of BRCA2 and other DDR alterations. So we perform next generation sequencing at the end of our assay, and what we get out is this sequencing data. We run this through our optimized pipeline, our bioinformatics pipeline.
Yeah.
I'm very impressed. Yeah.
No,
Am I going too far?
Fantastic.
No,
No.
We run it through our bioinformatics pipeline. What we get at the end is basically a list of pathogenic alterations that we see in our patients. And we also can estimate circulating tumour fraction. So that's basically the proportion of tumour DNA as part of the whole of the cell-free DNA that we get from the plasma. So once we have this data from our patients, we combine it with our clinical data that we get from the clinic, and we can really start to perform these association analyses and this statistical analysis where we can try and identify genomic markers that might be associated with either resistance at the beginning of therapy, they're not going to respond very well, or associated with acquired resistance. So they sort of their tumours may evolve over the course of therapy and they start expressing various somatic mutations that may mark them as becoming resistant to treatment. So in that way, that's basically how we go from an MCRPC patient presenting in the clinic. They get their treatment, we take their blood, all in the same building. We're doing all of this wet lab work, all of this bioinformatic work, and we get the data at the end, and hopefully apply it in the clinic.
Yeah, I mean, it's actually really incredible. I mean, I actually first started doing, when I was in Vancouver, 2013 to 2015, we were one of the first groups, probably there's only a couple of groups in the world then working on cell-free DNA and prostate cancer. And you know, then we were happy that we were able to pick up like changes in the androgen receptor 1 gene in plasma. We thought that was so cool, and we published a paper on that. And now we can do 89 genes with high, you know, sensitivity and specificity. And actually, what Heidi, you know, didn't say is that when you actually compare, you know, liquid biopsies to tumour biopsies now, there's very high concordance. So it actually is, it's not a full replacement, but it actually is a pretty good substitute for a tumour biopsy, which is obviously a lot easier to get. All men will give a blood draw.
Exactly.
But, you know, so
and that's exactly the thing. You know, tissue is a scarce resource.
Yeah.
And, you know,
hard to get.
Blood
painful.
is being collected on these clinical trial patients all the time. And the fact that you're able to use that to your advantage and kind of come up with the sort of techniques to extract what you need for your studies is amazing.
Yeah, I mean, we've looked at, you know, we found lots of genomic markers which correlate with clinical outcomes on, you know, novel hormonal therapies, immunotherapy, chemotherapy, and so now we're applying that technology to, or we will be applying that technology to Lutetium PSMA. So for the Lutetium PSMA registry and for all the clinical trials that we've got sort of upcoming or ongoing. We've got serial plasma samples that have been collected. So we're going to have, as we spoke about, we're doing these sort of world-first academic studies, which is great from a clinical perspective. And we'll talk about a couple more new trials that in a moment with Louise, but we'll actually have the opportunity to lead world-first translational research to sort of better optimize how we use these, how to use these treatments.
So Heidi, I mean, what for all our listeners out there, what's the big picture over the next 5 or 10 years? What do you hope to what's the buzz line, I guess?
In the context of Theranostics.
Of patients with metastatic castration-resistant prostate cancer and biomarkers. What do you hope to achieve?
I would love for them to become part of routine clinical practice, really. Even at the hormone-sensitive state, we know now that we can actually pick up cell-free DNA as well in our patients in the hormone-sensitive state. So I'd just love for them to be part of standard of care. We take the sample, we can actually process it in real time, so we can get enough information either at the start of therapy or over the course of therapy to sort of guide the treatment.
Fantastic, yeah.
Yeah, I think there's a really great opportunity, and I think we've also, one of the strengths, at Peter Mac is that we are all in one building, which most of the time is good because we get along most of the time. But the advantage is that as Heidi said, the sample comes from the clinic up to the lab. But we're also integrated with PET imaging as well. I think Michael Hoffman and his team have shown some really nice data recently, obviously showing that PSMA intensity is and FDG expression are sort of predictive and prognostic markers. Yeah. And so I think we've got the opportunity to integrate that with our genomic data, and perhaps we'll be able to come up with composite signatures of predicting clinical outcome or associated with clinical outcome. So I think there's some. I think that's a real
gap as well that we're all identifying is there.
Because even with in a way, Lutetium PSMA is a personal, is a sort of a personalised therapy.
Absolutely.
But even within that, as we see, as olaparib is, for BRCA1/2 patients, altered patients, but we know that even within that there's still heterogeneity in response and outcome, and that's where and cell-free DNA won't answer all the questions, but it might help answer some of the missing
even if
questions.
It is a question like what is a dose, what is the right dose to use for patients? We don't know the answer to that, and until we know the answers to these questions, that will be hard to answer. So yeah. Fantastic.
I mean, I think another thing, and this is, and I think it's a good opportunity to bring Luc in, is that we know prostate cancer is extremely there's heterogeneity is a massive problem, and there are so many signaling pathways and pro-survival pathways that are upregulated in context of ADT and all these sort of therapies, and so there's opportunities, I think, to bring in new combinations, and we've got a lot of targeted drugs now. So, Luc, what do you think are some of the what are some of the key pathways and targets that you're working on, or that you'd like to work on, you think could make sense to combine with Lutetium PSMA?
Very good question, Arun. I think I took a step back a few years ago when it comes to cancer heterogeneity. You mentioned personalized medicine, in which the idea to always look at specific mutation and how you could go and target these cancer cells that have these alterations. But at the same time, this heterogeneity also hides the fact that some cancer cell might share some commonality, some common drivers. And I think when we think combination, we have to think about the idea of combining therapies that can affect potentially all cells, no matter the heterogeneity, with the targeted therapy. And we can come back to, the known pathway, for example, the AKT, loss of PTEN, loss of RB, alterations in common oncogenes that drive very aggressive prostate cancer. But since this is, a theranostic preceptorship and PSMA therapy, I was thinking also it's just opening the door. I trained as a biochemist originally, and to see now the concept, with the FDA approval of PSMA 617 and lutetium therapy, that people can now bring, instead of only looking at functional target of that, because in heterogeneous tumor, people are looking at what are the drivers of all the cancer cell. But with PSMA, for example, PSMA doesn't necessarily, as a protein, have a main role as a driver of the cancer. It just happened to be there. And I think what's very exciting now is people discovering theranostic, young people are potentially going to go into that field, and then look at new targets, develop new peptides, combined with new radionuclides. And this can really open the door, as a good friend who's a medico and a researcher often reminds me, surgery and radiation were always, the most curative approach with cancer. And, one thing that's beautiful with Theranostics is, to basically bring targeted radiation. And I think that, that really opens the door to not only combination with known agent or agent we're developing against these pathways that are activated in CRPC, but also, as I said, potentially bringing completely novel target to prostate cancer with new Theranostics. So it's an exciting time.
It's we hear the nuclear medicine physicians talking about all these different radionucleoids, and it's like the periodic table. I thought that was like you just did something. That's just something you learn in year 10, and then it's like never go back to it. But suddenly it's like terbium, actinium. What is this? What is this stuff?
The lanthanides and actinides that are usually collapsed in the periodic table.
What?
Well, I actually saw gallium and lutetium on the periodic table the other day.
I'm going to have to look at it again. I think I'm going to have to look at it again. I think actually no one's actually put that. No one's put a picture of the periodic table on their slides. We should actually
idea for next slide.
Idea for next time.
But I think you're right. I mean, I think combination therapy is the future, and understanding the different mechanisms of resistance, developing those genomic biomarkers will really help us to determine which combination is best for who. Really is this era of personalised medicine.
I think so. That's actually a good opportunity now to perhaps ask Louise about her
trials. Yes, I'm really excited, Louise. Tell us.
Louise will be leading a couple of world-first combination trials that I don't think industry would ever do. And I think Tom Hope made a really good point today, didn't he, about sort of when he sort of juxtaposed the industry trials and the academic trials and how we take risks and have good control arms and do things that industry won't do because we have a different focus, right?
That's
It's not about making money, it's about answering important scientific questions. So,
correct.
Louise, do you want to tell us about your 2 trials that will be opening later this year and how they fit into the picture?
Yeah, sure. Thanks, Arun. So to start off with, we heard today at the Preceptorship how amazing Lutetium PSMA is, and it definitely is a game changer and has obviously recently been FDA approved. But the issue is that the responses don't last, even for exceptional responders. And even for the trial patients who had both a PSMA PET and FDG PET and had a suitable phenotype, some even have primary resistance. So there's definitely further research needs to be done in terms of why that is. There's sort of 3 main schools of thought in terms of mechanisms of resistance to lutetium PSMA. And the obvious one is that the prostate cancer cells don't express PSMA, and that's what we try and, I guess, weed out with the screening PET scans. But we obviously can't see micrometastatic disease on the PET scan, so that's one possible reason why it might not be effective. The other is that we suspect particularly the micrometastatic disease might not actually receive a lethal dose of radiation, and that comes down to the qualities of lutetium as a radionuclide. It's what we call a beta emitter, as opposed to other types of radiation people might have heard of, like gamma radiation, and that has a certain path length that can't reach single cells or small cell clusters, which is particularly present in the bones. And we often see patients progress on lutetium in the bones with developing more osseous metastases. And the third category is really intrinsic tumour characteristics, and that's to do with having DDR aberrations that might confer radio resistance. So they're sort of the 3 areas of resistance we're working on, and the trials that we've designed are trying to address 2 of those. So the first trial is called the LuCAB trial, and this is looking at combining lutetium PSMA with cabazitaxel. So it'll be really a 3rd-line study for metastatic castrate-resistant patients. Hopefully we'll open in a couple of months, so hoping maybe July this year. And the rationale behind the study is that patients will get the full dose of lutetium PSMA, but trying to target that potentially PSMA negative disease as well. But the good thing about cabazitaxel is that it's also a radiosensitizer, so in addition it may actually improve the effectiveness of lutetium PSMA.
Yeah.
So just before you go on to the next trial, Louise, I think one of the things there is, is cabazitaxel really underutilized in prostate cancer patients. It's like, I think the PBS data is like 10 to 15% of men get it. It's a life-prolonging therapy. So it needs to be used. And we know from the therapy trial where there was a cabazitaxel control arm, there were men who responded as they do, and there are men who responded to cabazitaxel even who hadn't responded to lutetium. So we're trying to sort of bring it together, you know, and leverage the radiosensitizing properties, but it's also a fact of actually getting treatment into these men. So it's a really exciting combination, and we hope, you know, it's a phase 1 dose escalation study, and if, you know, we'd like to then take that forward. But I think it's a really exciting combination. An important combination. But anyway, let's go to
fantastic. And the
next trial, which I think is even better.
So the next trial is focusing on the theory that potentially some of the prostate cancer cells aren't receiving enough radiation. So there's a trial called Alphabet. We've designed, Arun thought of the name. And
Alpha and Beta. Alphabet.
Wow. Did he insist on getting the accolades?
He insisted on the beta symbol.
Alpha, Beta, drop the A and just make the Greek symbol for B.
I get it. You know, I'm just going to interrupt there because Tom Hope did make a mention today that Australians are very creative at thinking of trial names. You know, therapy.
Yeah, I'm proud of that one.
Lutectomy. So he's looking to hire someone in the US. Are you interested? Go ahead, Louise. Sorry to interrupt you.
No, that's all right. So as the name suggests, this trial is actually combining what we call an alpha emitter with a beta emitter, which is the lutetium. So it's also a phase 1 study giving the standard dose of lutetium PSMA that was given in the Vision trial, but also combining it with an alpha emitter, which in this case is radium-223.
Yep.
And radium is actually already approved for use in prostate cancer. We don't actually use it really at all in the public setting. It's used privately at some hospitals.
It gets a fair bit of use in the US though, I must say. It does get some use in the US, but not in Australia.
Not in Australia. So the reason to combine these two agents is that we know that patients who progress on lutetium, as I mentioned, most commonly progress in the bones. And radium-223 is a calcium mimetic agent, so it almost acts like calcium, so it only acts on the cancer in the bones. It doesn't actually treat prostate cancer elsewhere in the nodes or viscera. But we hope that combining the two, that micrometastatic disease present in the bones will be treated. The difference with radium and alpha emitters in general is that it has a higher, what we call linear energy transfer, which means it sort of gives more bang for buck. There's more energy in the actual radiation, and the path length is much smaller, so it's able to reach those tiny cells.
So you're hoping that by
She sounds like a new
I know, it's crazy.
Tell Hoffman to watch out. I mean, seriously, like, listen to her.
So you're hoping that by using both agents, the alpha and the beta, it's going to reach those sites that may be resistant to just beta.
That's exactly right, in the bones.
Yeah,
and
fantastic.
Looking at the toxicity of both of those agents on their own, they're both generally well tolerated, particularly radium-223. So we're hoping it will be well tolerated, but we will just, to be safe, and because it is a first in human combination, we will follow a dose escalation design with the radium to start with.
Yeah. Fantastic.
And I think the other thing is to then link back to some of Heidi's work is that there's serial plasma collection for the cell-free DNA incorporated in this work. So that'll we'll develop a new combination, hopefully show it's safe in both trials, and then that can then move forward in clinical trials. But we're also generating the translational data at the same time, and some of those new targets that maybe then Luc can work on with some of his other preclinical models. So I think it's it's hopefully giving us more than just clinical trial data.
And it's great that none of these trials are sort of mutually exclusive. I think what you find from one is going to directly impact the other. And I want to tell you about my PhD, Arun.
Yes,
you want to hear about it?
Well, that's right. Well, that's exactly right. Well, this all fits in. So tell us. Yeah, of course. You're the host, so you can do whatever you want.
I can do whatever I want,
right?
No, but what it got me thinking is that the Lutectomy trial, and we've got a couple of upfront PSMA trials. Your PI on one, and Declan Murphy's PI on the Lutectomy trial. And the beauty of this trial is, we've talked about how scarce tissue is, but this is a trial where we take biopsies from pre-treatment biopsies, and then
localized prostate cancer.
They're getting upfront lutetium PSMA and then going on to have radical prostatectomy for their localized prostate cancer. And the beauty is that we get the tissue from before they have their treatment in the biopsies, and then we get the tissue from after their treatment. And we really get to have, we get a great insight into what's happening in the tumour microenvironment.
Yeah.
And, like you said, prostate cancer it's a very heterogeneous disease, and so far that is what we're seeing, very heterogeneous results. But understanding what happens in the tumour microenvironment in response to these therapies will, it will really shed a lot of light on what are the resistance mechanisms,
Yeah, no, it's absolutely right, and it's interesting because, and I've had this discussion with Declan, numerous times about neoadjuvant androgen deprivation therapies or hormone therapies have been used a lot in prostate cancer, right? And they've never really penetrated because the responses are never great, and there's never really pathological complete responses. And the biology always is, there's still androgen receptor signaling, and there's still tumour there, but I don't know that they've looked at it from an immunology perspective. And I think that's what you're doing with Paul Neeson and co,
right.
sort of looking at that and the sort of radiation biology side of things. So I think there's it's a really fascinating, opportunity, and we have to mention this, of course, that when we presented this work to Howard Sewell and PCF, I mean, he was extremely excited, and of course that led to you having it was a Zoom call with a Nobel Prize winner. I think
very exciting.
I think we need to share this with the audience. Have you shared this? You must have shared. Have you shared this with the audience before?
I don't think so.
Oh, so this is why I'm in the chair today. and Declan's been pushed aside because I'm bringing this stuff out. So.
That was a very exciting day. So we had a Zoom meeting with Jim Allison, Nobel Prize winner.
What year? 2019? Anyway, I should know that.
2019,
2019 was it? Anyway,
we'll
say 2019. Anyway,
we will confirm that.
Pre-COVID.
Pre-COVID world.
And Dr. Pam Sharma, both based out in Texas. At the MD Anderson Cancer Centre, and it was just, we were just really, Howard thought it would be a great idea for us just to chat. They've done so much work in cancer immunology, and we presented, what we were thinking and what our plans were for the project, and it was great to get their insight. And get this, it wasn't just them two; they brought their whole immunology team on the Zoom meeting. It was fascinating.
You must have a big lab.
Oh, I mean, both of them. They're fascinating people, and as immediately there were questions. Are you seeing CD3 cells?
Yeah.
Well, and then they would just ask one of their lab members, what can you guys do? Can you do something? And they'll be like, yeah, we can do something.
Yeah, I mean, so Jim Allison's the guy who discovered CTLA-4, right? And then ipilimumab came from there, so transformed cancer medicine. So, no, I mean, that's fantastic. But I think that shows the interest that people have got in understanding. And so I think there'll be really fascinating insights that will probably also translate to the metastatic setting, but it's almost impossible to, not impossible, it's much more challenging to do that in the metastatic setting.
Absolutely.
Just because of getting tissue. I mean, we do have, Belinda Parker and her team at Peter Mac we are going to get some biopsies from the Alphabet study so she can look at some of these, some of the tumour microenvironment changes and
I mean, I think
in the metastatic setting, but it's obviously much easier to do in the
That's right. I mean, the main advantage of this trial is really the access to tissue, before and after treatment. And, I think the phenotype, I'm sure, changes when it's metastatic castration-resistant prostate cancer, but still.
You say translational, but I had a urologist come up to me today who basically said, Arun, looking at Lutectomy, I think in 5 years radical prostatectomy is going to be obsolete because we're just going to.
I think I know who this is.
We're going to give Lutetium PSMA. If he's got a 2 millimeter tube, why wouldn't it just be melting away with Lutetium? I said, I think it may be part of multimodality therapy, but I don't think it'll replace radical prostatectomy. But it did make me think about Declan's talk about obsolescence, and that's why Declan is doing his sabbatical in nuclear medicine. He's one step ahead. So
he's thinking big picture, 10-year plan. Yeah,
but absolutely. I mean, it will be interesting to see where that goes. But I think the translational.
I think it's the translational understanding we can get out of this trial that's going to be really key in informing future trials and developing combination therapies.
Yeah, I agree completely.
Amazing. Gosh, these are spectacular women. And Luc, amazing person too. You guys are overpowered today. That's rare.
Yeah, that's good. No, that's very good. I saw we saw Shelley Dolan, our CEO, tweeting and saying,
another great woman.
Yeah, another great woman, and Professor Shelley Dolan, I should say, and she made comment on Twitter like the panels, for the preceptorship were really well balanced with gender and age and also culturally diverse. So I think it was really a credit to Michael and Annette and Liz and everyone else who's put together the meeting. Yeah. It's actually really important. And
I think it's testament to the fact that, we're all like-minded in this way. And this, it's the future of conferences is this.
Yep, for sure.
Well, it's been fantastic to have you here. We could talk about this forever.
We could go forever, but we can't.
So many exciting things happening, and we will be back hopefully tomorrow with another podcast. There's so many superstars here. Arun, you might have to join me again.
I'm expecting a tap on the shoulder.
Excellent. Oh, by the way, we found these two at the bar. Is that saying something about you as a supervisor? Are you driving them to drinking?
Yeah, well, I'll step out of the room and then you can ask them that question again. For the next few years, that's right. Heidi's giving her the talk. So.
All right, well, that's it from us, and we'll see you soon with another episode of GU Cast.